Hi, my name is Roumen Bogoev, and I'm head of segment management in GenScript. And it is my pleasure today to present to you about the GenScript advanced solutions for bispecific antibody development for cancer therapeutics. So the agenda for today's talk will be the following. First, I'll give you a little bit information about GenScript in case you're not familiar with the company. Then I'll discuss a little bit more of GenScript's portfolios for different therapeutic development solutions. And then I'll introduce you to the GenScript bispecific antibody solutions, what we offer stage by stage. And then I'll show you a summary of key services and products that we offer. So GenScript is a global company. We operate in many countries in the world. We have multiple manufacturing sites around the world. We have 2 manufacturing sites in US, one in New Jersey and one in Seattle, Washington. We have multiple manufacturing sites in China. We have regular production and GMP production. We also have GMP production site in New Jersey. We also have other production sites, for example, in Singapore, where we produce do a lot of the services to service our Asian Pacific customers. And we have many commercial offices around the world, including United Kingdom, Netherlands, Korea, Japan, and others. So the GenScript group consists of several entities. I'm not going to cover all of these entities today. I'll start from the right side because those are the entities I'm not going to cover today. Bestzyme is our synthetic biology company. They have very strong synthetic biology capabilities that they use for focusing on human and animal food supply. They basically have very large capacity. They produce enzymes and other products in tons rather than milligrams and grams as we do for science. Then it's Legend Biotech. Legend Biotech is now was spun off as a separate company a few years ago and is trading publicly on the New York Stock Exchange. They're an innovative cell therapy company delivering therapies for a range of disease. A few years ago, they launched Carvykti, working with Janssen for multiple myeloma. And they have advanced pipeline making progress on various therapeutics for leukemia, solid tumors, and infectious disease, mostly focusing on cell therapies. So these are the entities I'm not going to cover in my talk today. I'm going to focus on the entities on the left side and showing the capabilities we have and how we can service you for your therapeutic developments. So on the left side is, very left is the life science group from GenScript. That includes our CRO and product divisions. They offer a lot of reliable services and products with therapeutic development. There is a lot of automation being used in our CRO division to increase our efficiency, and we offer a lot of cutting-edge solutions. We also offer a lot of products for automating various processes like plasmid purification, antibody purification, protein purification, and so on. Our ProBiobiologics CDMO division focuses on basically preclinical and clinical work for customers. They have large scaled up GMP capacity to meet the market demand. They have very strong regulatory experience working with number of customers and everything from IND filings all the way through clinical 1, 2, 3 and commercialization. They're one of the leading biologics CDMO service providers in the world. So I'll focus on the capabilities for these 2 divisions and show you what we can do for you if you're developing antibody therapeutics and more specifically bispecific antibodies. So GenScript doesn't offer only solutions for bispecific antibodies and antibody therapeutics. We have very strong capabilities in a number of different areas. We have very complete portfolio solution for gene therapy development. We have for gene editing, virus delivery, non-viral delivery, and so on. We have very strong solution for cell therapy, starting with cell isolation, cell engineering, cell formulation, activation, formulation, and so on. Of course, antibody therapeutics, I'll expand on this a little bit later. We also have very strong capabilities for vaccine development mRNA vaccines, DNA vaccines, protein-based vaccines, peptide-based antigen vaccines, and so on. We also work with a number of customers on IVD developments, so we have very strong portfolios for molecular diagnostics or immunodetections. And now more specifically for bispecific antibody treatments for oncology. Bispecifics are very attractive methodology for developing cancer therapeutics because they have the capability to bring the T cell in close proximity to the cancer cell and basically facilitate the cell death for the cancer cells. There are a lot of surface markers that are being used for developing bispecific antibodies. Here on the left side, you can see the chart, the listing of different surface markers that are being used on the cancer cells, and they're targeted for different cancers. Here on the right side are listed the number of different cancers that you can target using these ligands. And on the right side, there are different bispecific antibodies that have already been launched as therapeutics. You can see that the majority of those are for oncology. So bispecific antibodies are definitely great methodology for developing cancer therapeutics. Of course, bispecific antibodies have a lot of challenges and a lot of considerations need to be taken. Basically overcoming the development challenges, starting with selecting the appropriate antigens, tissue or cell, providing data for the safety and efficacy, analyzing the mechanism of action. So there are a lot of challenges in developing bispecific antibody therapeutics. And these are some of the considerations one may need to take into account when developing bispecific antibody therapeutics. Basically selecting the appropriate tumor-associated antigen, something that is highly expressed in tumors and minimally expressed in normal cells, in order to ensure the increased efficacy and less toxicity of the drug. Of course, antibody engineering is used a lot, engineering the different types of molecules that you may need for your bispecific. We have multiple technologies for that. We have single B-cell technology that we can utilize. We have phage display technology. We have traditional hybridoma technology that we can use for developing antibody. And then we have very strong engineering capabilities I'll mention a little bit later for engineering this antibody to your specifications and optimizing your lead for further work. Of course, a lot of in vivo testing needs to be done to confirm that the antibody is engaging the T cells and also accordingly the cancer cells. And then a lot of preclinical and clinical development, successful in vitro candidates are tested in different animal models, and potentially you would go to clinical 1, 2, and 3 trials. So GenScript has very strong solution, very complete solution for bispecific antibody development. Even though the antibody development takes, it's a very complicated process and takes a lot of manipulations and has a lot of stages. First, you would start with target identification, then target validation, target preparation or antigen preparation for your antibody generation, then generating the antibody using different methodologies, antibody screening to find specific leads that work, then antibody engineering to further optimize your leads to your specifications, stable cell line development, preclinical studies, IND filings, FDA approval, and then clinical development and clinical manufacturing. So for all these stages, GenScript can offer very complete portfolio of solutions starting from beginning to the very end. So this is a kind of listing of most of the services and products that we have for the different stages of the development. I'm not going to go through all of them right now. I'll just point some categories. So for target identification and validation, we have very strong portfolio for genome analysis. We have protein analysis portfolio. We have cell-based analysis, cell-based assays. For lead generation and optimization, we can start from everything, starting from gene synthesis, various methodologies for gene synthesis, plasmid preparation, antigen expression, antibody development, functional testing of the antibody with various assays, further optimization of the antibody with various mutagenesis technologies and mutant libraries, and so on. Then candidate validation and IND studies, basically going to preclinical work. We have our CDMO division can offer very strong capabilities for that. Complete biologic CMC for IND. They have a lot of also separate services that they can offer. The pro-LVV lentivirus manufacturing services, the pro-AAV manufacturing services, the pro-COD cell line development services, lenti-helper plasmid, pro-plasmid manufacturing service, and so on. And then also on the CDMO side, we also have basically complete GMP manufacturing service, and we offer all the necessary analytical services. We offer GMP cell banking, GMP cell line development, and so on. So everything from beginning, from target discovery to lead generation optimization, through candidate validation, and then through clinical trials and GMP manufacturing, we can offer that for you through our two divisions, the CRO product division and the GMP CDMO division. So here are some of the more specific offerings we offer, especially our gene titan technology is technology for producing a lot of oligos, basically producing thousands and millions of oligos on CMOS microchip. And we can produce, one of our chip allows us to produce 92,000 oligos at the same time, and the other chip allows us to produce up to 8 million oligos at the same time. So we use these technologies for various library developments. We can do, for example, NGS enrichment libraries. We have some hybridization-based enrichment libraries, some amplicon-based enrichment libraries. We use them also for a lot of our CRISPR-RNA libraries. And basically very strong capabilities. We can very efficiently and very quickly produce various libraries using oligo pools. This is one case study working for a customer. They wanted about 50,000 gRNAs for knockdowns and knockouts. And basically, we produced the library with our gene titan technology. We produced the oligos. We cloned the oligos into the plasmids. And then the produced library, plasmid library, is QC'd. And basically, we do very extensive QC for that. We use NGS to sequence 30 random selected oligos to confirm that the sequences are correct. And we have very good coverage. We have 100% coverage with single gRNA, and we have 99.95% with 10x gRNAs for these targets. So this is just one example of our capabilities. We have also Cas enzymes. Basically, we have very strong CRISPR capabilities, everything from enzymes to gRNA and the various gRNAs, HDR templates that are required if you are doing knock-ins, for example, and so on. On the enzyme side, we offer a lot of different varieties, wild type Cas9, ESP Cas9, which is with much higher specificity. We have also Cas12a, Cas13a. We also offer some of the novel modalities. We recently licensed from Broad Institute. They're prime editors. So we have a variety of prime editors. And we also have some base editors. So very, very strong CRISPR capabilities. We also offer these enzymes in protein and mRNA format. In some of the mRNA formats, they're also prepackaged in LNPs. So if you go to our website, you can see some catalog products that are basically protein format enzymes, also mRNA format enzymes, and also LNP format enzymes. We have very strong expression capabilities. We have a number of different platforms that are typically suitable for different applications. We have bacterial platform, insect platform, mammalian cells platform, cell-free platform. They have different advantages, and we typically use our bacterial platform for producing any kind of enzymes, antigens, cytokines. We use our insect platform for soluble proteins, membrane proteins, complex proteins. We use our mammalian platform, either CHO or HEC293, for various antibody formats and secreted proteins. And we use the cell-free platform, which is new offering for us for high throughput, basically AI-driven developments. If you want to develop thousands of samples, for example, VHHs with tags or without tag, in other formats, we can do them with the cell-free platform. We have capabilities of 1,000 samples per day. So we also have put a lot of effort into our turbo-CHO expression platform. We have developed several different variations of it, and we have optimized it to be very efficient, high throughput, and very fast. For example, our turbo-CHO high throughput platform, we can produce antibodies up to 10 milligrams each, and the service can start from 5 business days. Basically, in 5 business days, we can produce the antibodies. Starting from sequence, we can synthesize the genes, clone them into vectors, transduce the cells, basically express into mammalian CHO cells, and purify the antibodies within 5 business days. We also have the turbo-CHO Express, can produce from 10 milligrams to 1 gram per target. That takes about 2 weeks, starts on 2 weeks, depending on the amount of antibody that you need. And also turbo-CHO high performance from 1 gram to 200 grams, usually for more like IND studies and clinical 1 studies in some situations. Basically, that takes about 6 to 7 weeks. We do, of course, very high level of purification. We offer different assays for the different services. Very often we offer the purity, protein purity analysis, endotoxin levels, and target concentration is usually at least 1 milligram per milliliter. We also have these other assays that are optional here that you can add if you prefer. We have ELISA assays, cell-based assays, FACS assays, octet, BIACORE, SPR assays, and so on. And you can see on the chart below, basically, with our turbo-CHO high throughput, you can start with your discovery studies, then lead generation. Usually you can use our Express turbo-CHO, and then your candidate selection, further testing. You can use our turbo-CHO HP high performance platform. And then after that, of course, we can work with our CDMO division to develop your stable cell line and proceed to clinical studies with that stable cell line. Here is just a little bit more information about our turbo-CHO platform. We have done many different bispecific antibodies with this platform. We have done over 70 different formats. We have done over 5,000 projects. We have over 10 years of development experience. We have optimized the process very specifically for antibody production. And on the right side, you can see basically different antibody formats that we have done. We have done fragment formats, symmetric antibody formats, asymmetric antibody formats, and so on. And the barcode here can show you some more information about, if you want to know more about our turbo-CHO capabilities for bispecific antibodies, it can take you to the link for with that paper. Here is another case study developing bispecific antibody. Basically, we develop the antibody and produce it, and it's a human IgG G1 like bispecific antibody. You can see the structure there, the format, and we produce it with very high yield, with 374 milligrams per liter yield, and very high purity, SDS-pH at least higher than 95%, and SEC higher than 99%. Precision mutant libraries, as I mentioned, we can use our Gene Titan technology to produce a lot of different precision mutant libraries. The advantage of our libraries are that they're high quality, very diverse synthetic libraries that contain only the desired mutants. We have precise control over the incorporating of the individual nucleotide into the basically the oligo, even distribution of the amino acids with very little bias, inclusion of user-defined codons, elimination of any kind of stop codons, and we can perform also codon optimization for the library. And basically, we can produce site saturation libraries, saturation scanning libraries, combinatorial libraries, and so on. And we can produce them in plasmid or lentiviral vector formats. Here is a case study producing combinatorial, basically precision library compared to traditional NNK library. Here on the left side, you can see the distribution, amino acid distribution for the NNK library. On the right side, you can see the precision mutant libraries with much better amino acids distribution. And then you can see on the right side, significantly increased efficiency for the precision mutant library compared to the NNK regular traditional library. In addition to the services, we also have a lot of off-the-shelf reagents. For example, we have many stable cell lines that are off the shelf that you can just purchase and they can be delivered to you within a couple of weeks. We have Fc receptor cell lines for antibody characterization. We have GPCR cell lines, very often used for small molecule drug development. We have immune checkpoint cell lines, obviously very applicable to cancer research. We have Claudin, CODN cell lines. Claudin is becoming more and more prominent target for a number of therapies nowadays. And we even have some viral libraries like ACE2 and Spike COVID libraries, COVID-related libraries. We also have a lot of off-the-shelf antibodies. I only point some of the categories here. We have a lot of very good anti-VHH antibodies that you can use for detecting your VHH candidates. We have epitope tag antibodies. We are one of the biggest vendors on the market with one of the biggest libraries of anti-U type antibodies that are off the shelf. Of course, we can also develop some. I'll mention that to you later. We have a lot of reagents for immunodetection and IVD antibodies also. We have a lot of in vitro bioassays. We have platform that can develop assays in 14 different categories: growth inhibition, GPCR agonist, antagonist, CDC, ADCC, cytokine neutralization, FACS, other assays. Basically, we can develop custom assays. We also have 40 ready-to-use assays for various targets that are off the shelf. But basically, we have very strong expertise in various bioassays that may be needed for any kind of biological development, including bispecifics. Here is a case study developing bispecific antibody, basically bispecific antibody to two targets, for one BB and tumor-associated antigen. And on the left side, you can see a very nice titration curve when we use CHO K1 cell line with tumor-associated antigen target displayed. And we use the 4-1BB reporter, basically reporter ligand. And you can see very nice titration curve for the bispecific antibody. On the right side, when we use the CHO cell line, regular CHO cell line without the target displayed, you can see there's almost no significant non-specific binding for this antibody to the CHO K1 cell line. We have very strong bioinformatics platform, and we can support your developments from beginning to end. Basically, we have this bioinformatics platform allows us to do epitope prediction. Then the next stage for antigen preparation, we can do codon optimization. We can do expression prediction. For the NGS sequencing, of course, we can do the NGS analysis, and we can do variable domain sequence analysis. Then for the developability assessment, we can do variety of analysis, immunogenicity prediction, viscosity prediction, PTM sites prediction, aggregation sites prediction, degradation sites prediction, developability analysis report, monoclonal antibody chain domain annotator, and bispecific antibody structure annotation. And then for antibody engineering, of course, we can do humanization, colonization, felinization, binding antibody maturation. For the antibody production, we can do codon optimization, expression prediction, and then for QC, we can do a lot of analytics, gene electropherogram analysis, protein purity analysis, protein mass spectrum, and so on. So very strong informatics platform that can support you for your developments. In these here, the ones that are with the rectangle, developability assessments, they can be added to your TurboCho smart quote. They're available there for selection and basically for addition to your service. Anti-type antibodies, we can develop, as I mentioned, we are one of the biggest vendor of anti-type antibodies with the biggest off-the-shelf selection, but we can also develop a variety of anti-type antibodies. We can develop blocking anti-type blocking antibodies that detect the free antibody. We can develop non-blocking antibody that detect the total antibody. We can develop drug complex specific antibodies, basically that will detect only the bound target to the antibody. And basically, we can also develop a full assay for you. We can develop a lot of your pharmacokinetics and immunogenicity assays in addition to just developing the antibodies. Here is just a case study for developing anti-type antibodies for trispecific antibody. We did immunization, and basically we selected 133 clones of SCAV, his fragments. Then top of these 96 are blockers, and with blocking rate of above 30% with blocking ELISA. And we selected the top 3 and tested them further with for antibody ELISA. You can see here on the right side, the bottom chart shows very nice titration curves of the inhibition results for these 3 selected anti-type antibodies to the trispecific antibody. And this is just summary of some of our key services and products, just to give you overall understanding of the portfolio that GenScript can offer. You can see that we can offer services and products from beginning to the end, from target identification, lead generation, preclinical, all the way to IND and clinical trials and production, antibody and protein expression, various grades of services, as I mentioned, for the TurboCho, as well as other platforms, all the way through the IND and GMP grade. CRISPR, gRNA, and HDR templates. Also, we can offer research grade, GMP-like grade, and also GMP grade for clinical trials and production. Plasmid preparation, we offer research grade, industrial grade, preclinical grade on the research side. And then we have IND, CMC, GMP Pro, and GMP grade for the clinical studies and manufacturing. IVT mRNA, we can also offer research grade, preclinical grade, and GMP grade. AAV vector, REO services, AAV Pro, and then AAV CMC, clinical and GMP grade. Lentivirus also, complete capabilities from research all the way through GMP. Peptide synthesis, of course, we have very strong synthetic peptide capabilities, research grade and GMP grade. We also offer a number of very unique and innovative products. So we have excellent system for For cell isolation, we have some products that can be used for research use that are manual, and we have fully automated platform for research use or for GMP use. And that platform also we can supply with nanobits with associated with the selected antibodies for them to select for, let's say, T cells, NK cells, stem cells, and other cell types for your cell therapy research and development. We also have excellent T cell activation reagent that we offer in research grade as well as GMP grade. And we have also plasmid purification system. So this is fully automated system for purifying plasmids. We have two systems. One system is for mini prep, can do up to 48 mini prep samples at the same time. And the other one is for maxi prep / almost mega prep, milligram quantities of plasmids up to 6 samples at a time. All of them fully automated from beginning to end. You just add the cell pellet, and the system will do the rest and give you purified plasmid, transfection grade purified plasmid. We also have protein purification system that can purify anything that is with a tag, basically any tagged protein or antibodies, and can do it in high throughput and very efficiently using magnetic beads. So this is kind of overview of some of the key services and products that GenScript can offer for you. And of course, we have serviced many customers through the more than 20 years of GenScript's existence since 2002. And we have hundreds of thousands of publications in peer-reviewed journals, some of them in Cell, Nature, PNAs. You can see the increasing number of publications every year. So we support a lot of our customers in their development through different stages of the development, and we support them with their research. And thank you for your attention. This is my basically last slide. Thank you.